Welcome to lesson 2.6.3 on cell division. This is also can be found in your textbook in 10.2. One of our two overriding questions today is what events occur during each of the four phases of mitosis. So most of you know that the four phases are prophase, metaphase, anaphase, and telophase, but what are the events that are going on there? So there's some vocabulary here that you need to know that in order to understand all of this. Chromatid means each strand of a duplicated chromosome. Centromere is where each of those chromatids are joined together. Centrioles are tiny structures located in the cytoplasm of animal cells that help organize the spindle fibers. And then spindles are fan-like microtubule structures that help separate the chromatids. Make sure that you know those definitions. We're going to be using these words as we move along. Alright, so let's get into it. The first step is prophase. And during prophase, the first phase of mitosis, the duplicated chromosomes condense and become visible. So our chromosomes appear. Our chromosomes condense and appear. Now remember, prior to this, our DNA was in the form of chromatin, which was too thin to see. So we see our chromosomes here that have appeared, and so the next thing that happens is we have these centrioles, and the centrioles are organelles that were made during G2. And so the centrioles will move towards the poles. They'll move to the poles, and with them come these spindle fibers. And what they're doing is they are organizing the spindles. These spindles are like bungee cords, which our chromosomes are going to attach to. So here are our spindles. Our spindles form, and the DNA strands attach at the point called the centromere. So now our chromosomes are going to attach to one another, or our sister chromatids are going to attach to one another at the centromere. And eventually these spindle fibers are going to attach to our chromosomes at the centromere. The last thing that happens is our nuclear envelope disappears. So we're going to have the chromosomes appear. They're going to attach to one another at the sister chromatids are going to attach to one another at the centromere. We're going to have the centrioles start to move towards opposite poles of the cell, and our spindles are going to form. And finally, our nucleus is going to disappear. Our nuclear envelope is going to disappear, and we're no longer going to see it. In metaphase is the shortest phase of cell division, shortest phase. And it is very clear, and you should have seen during the lab, that this is the clearest phase of cell division. And what's happening during metaphase is the centromeres of the duplicated chromosomes are going to line up across the equator of the cell. So chromosomes line up. And this equator is actually called the metaphase plate. So if we drew a line through our centromeres here, we have what we know as the metaphase plate. We also have the spindle fibers starting to attach to our chromosomes. So spindles attach to the chromosomes. And they will attach at the centromere, but at the centromere there's like a little belt here that is called the kinetochore. Kinetochores will attach to the spindle fibers, and this is all kind of right at that centromere point. Right, so we've got this metaphase plate forming, and this is going to be an extremely short phase because as soon as we get all of our chromosomes lined up, they're going to start to split apart. So here are our spindle fibers attaching to the chromosomes, and then in anaphase, anaphase begins as soon as we start to split those sister chromatids apart. So during anaphase, the centromeres are going to be pulled apart and the chromatids separate. So chromatids separate. And anaphase is going to continue until we get all of these chromosomes to our opposite poles. So the chromosomes are pulled, let's say pulled instead of moved. They are pulled by the spindle fibers to the poles. And this is going to ensure that we get equal DNA. Remember that these two chromatids are exact replicas of one another. So each new cell needs a chromatid, each of the chromatids. So they're going to be pulled apart, and this all ensures that we're going to get the same exact genetic information in each of our new cells. In telophase, we've got our chromosomes at the poles, so our chromosomes are at the poles. Our nuclear envelopes are going to reform, so we're going to reform our nuclei. And our spindle fibers are going to disappear. And telophase is going to kind of mesh in with anaphase and with cytokinesis. So we're about to talk about cytokinesis, but cytokinesis starts during telophase. They are two different processes because remember, mitosis is the splitting of the nucleus, which is essentially what we've done here. But cytokinesis is going to start during this telophase portion. And we can see that we start to have this indentation, this pinching action, which is going to separate these two new animal cells. So, the events that occur during each of the four phases of mitosis, we have prophase, in which our DNA is going to condense and become visible, and then our nucleus is going to start to disappear and spindle fibers form. During metaphase, the centromeres are of the duplicated chromosomes are going to line up across the center of the cell, and the spindle fibers are going to attach to the kinetochore at the centromere. During anaphase, the chromosomes are going to separate and move along those spindle fibers to opposite ends of the cells, and then during telophase, the chromosomes, which were distinct and condensed, are going to begin to spread out into a tangle of chromatin, and so we're going to start to lose our chromosomes. And I forgot to write that down, but remember this part of it. We're going to move from chromosomes to chromatin. We started prophase as the chromosomes became visible. Now they're going to disappear. So our next question is, how do daughter cells then split apart? We split apart the nuclei, but we now need to split apart our cells. And this is different in animal and plant cells. In animal cells, the cell membrane is going to be drawn in, and there's going to be this pinching action. And the cell is going to pinch into two equal parts. All right, so we've got a cell, and we've gone through anaphase, so our chromosomes are down here. Well, there's going to be a pinching action here, creating a cleavage furrow. I didn't do that very well. It would be something like this. And we're going to start to form our nuclei. This pinching action here is called the cleavage furrow. Cleavage furrow. And it's going to, so there's supposed to be two R's there. This pinching action is going to continue until we separate into two new cells. So we kind of go in this action here. In plant cells, because the cell wall doesn't allow us to do that pinching action, we're going to form what's known as a cell plate. So after anaphase is done and we've got all the DNA down here and we start to form our nuclei, what's going to happen is we're going to start to form this cell plate right in the middle, and eventually that's going to form our two new membranes, and we're going to get a new cell wall between the two cells. Another important distinction between plant and animal cells in mitosis is that there are no centrioles. In plant cells. We do have spindle fibers, but no centrioles. So, in cytokinesis, we complete the process of cell division by splitting one cell into two. Mitosis, we've split the nuclei. In cytokinesis, we've split the cell. So my last slide here is kind of the stages of the cell cycle. We start with interphase, in which we're going to grow, we're going to replicate the DNA, and we're going to prepare for cell division. We go to prophase, our DNA is going to become visible. We're going to start to form the spindle fibers, and our nuclear envelope is going to break down. Metaphase, we're going to line up all the chromosomes on the metaphase plate, and the spindle fibers are going to attach to the kinetochores, which are located at the centromeres. Then in anaphase, we're going to split our sister chromatids apart. In telophase, we're going to start to undo prophase. We're going to lose our chromosomes. They're going to become chromatin again. We're going to form a new nucleus, and our spindle fibers are going to disappear. And in cytokinesis, we divide up the cytoplasm into two equal parts, either by a cleavage furrow or by a cell plate. That's the end of the lesson. Make sure that you know what happens in each step as we move along, and I will see everybody tomorrow.